All right, let's take a look at cell division and the division of somatic cells. So essentially, when we're talking about somatic cells, we're talking about all the cells of the body except the gametes, G-A-M-E-T-E-S. Gametes are the sex cells, the sperm and egg cells. So we're talking about the cell division that would occur, or the cell cycle that you would have for skin cells, muscle cells, epithelial cells, nerve cells, and so on. The cell cycle is going to be defined by a series of stages, and those stages each have something in particular going on, something that defines that stage. So it's predictable. If we say that a cell is in a given stage, a biologist will know exactly what is happening with that cell at that time. If we look at the whole cell cycle, we have interphase here represented by the green arrows. Interphase is when the cell is doing its normal business. It also includes a period of time where the cell is dividing. And then, as you see indicated by this darker orange arrow here, we've got a portion of that cell cycle consisting of mitosis. And mitosis is the nuclear division of the cell. It's when the cell is actually shuffling the copies of DNA to each pole of the cell, so that when it divides in half, each daughter will have a full set of the original DNA. And if we look at that with a very similar diagram, you can see here that interphase consists of three substages, G1, S, and G2. You look at this blue arrow in the center. That shows you that all of this is interphase. About 90% of the cell cycle is spent in interphase. It depends on the cell, it depends on the species you're talking about, and the conditions, but the bottom line is the vast majority of the cell cycle is spent in interphase, and then, as you can imagine, a relatively short period of time consists of the actual division of the cell from a parent cell to what we refer to as the two daughter cells that are present as a consequence of that division. And if you look at mitosis, this little yellow piece of the pie, even more closely, you'll see that mitosis consists of these four key phases. I define 5 for you because I included this transitional phase not represented in the diagram called prometaphase. But essentially we've got prophase, metaphase, anaphase, telophase, PMET. And if you look at what's going on in each of these, it's always going to be the same thing that occurs at each stage. It's always going to be the case that something in particular is happening in prophase, something very particular is always happening in the same way in metaphase, anaphase, and telophase. And that's why those stages can carry those names, and those names have meaning regarding what's happening with a particular cell. And then cytokinesis here refers to the division of the cell. Of course, kinesis refers to movement, cyto refers to the root word, refers to cell. So this is the actual separation of the two daughter cells into two separate entities. G0 here might be a little bit confusing, but this is indicating that, hey, cells can actually exit the cell cycle in the sense that they can stop dividing. And so you have certain cells that enter that state, and we can get to that a little bit later. First, we want to understand what mitosis entails and what's going on in interphase. So most of the cells that we're talking about are doing their business in the sense that they're performing the regular functions throughout most of their existence, but they'll spend, depending on circumstances, a short amount of time dividing by mitosis. Mitosis is what we're emphasizing here, so you understand how cells divide, but of course there's going to be great variation in terms of what's going on with the cell during interphase, and that depends on what that cell is. We have all sorts of cells in our body, for instance, as multicellular organisms. We have all sorts of different kinds of cells performing different jobs. We have epithelial cells that might be the lining of our gut, it might be our skin. We have nerve cells. We have different kinds of neurons. We have all sorts of glial cells, which are cells that exist that support the nervous system, such as oligodendrocytes and astrocytes, and we have microglia. We have all sorts of cell names that you've probably never heard of before. Even if you know that we have blood cells, for instance, we've got red blood cells and white blood cells. And if you look at white blood cells, we've got eosinophils and basophils and dendrocytes and helper T cells, cytotoxic T cells, plasma cells, B cells. So the list goes on and on. Even our skeletal system has different cells. You might see something like human bones as static, but no, they're living tissues. It's very dynamic, and we've got all sorts of different cells involved, even in that tissue. Muscle cells, skeletal muscle, cardiac muscle, smooth muscle. The list goes on. And so what's common to these is that cells do divide, so there'll be a little bit of cell growth, but most of our growth occurs by adding cells, not by expanding the few cells that we have. So we went from one fertilized egg, each of us, and other organisms go from one fertilized egg, if you're talking about eukarya, to or if you're talking about multicellular eukarya, we go from one fertilized egg to 2 cells, to 4 cells, to 8 cells. If all 8 cells divide, of course, we get 16 cells, and it goes on and on and on. And so, in any case, you get the idea. Growth and development, we've already discussed, that happens by mitosis. For some asexual organisms, mitosis can actually serve in reproduction as well. We're looking at animal development, going back to talking about, say, ourselves, or polychaete worms in the marine environment, or butterflies, whatever you want to discuss here. We've got, as an example, we've got fertilized egg. They skipped the 2-cell and 4-cell stage. We've jumped to an 8-cell stage here, and of course there'll be a 16 and many other stages between that, and hundreds of cells here we have as a blastula, hollow ball of cells, that then eventually develops into what we call a gastrula, and an embryo with more than one tissue layer. We discussed this in class a bit. Remember, we're not studying animal development. You don't have to worry about this slide. It's just to say that, hey, we're growing by adding cells, and eventually we can get into some interesting stuff that does involve development, and we can chat about how cells become different in the first place. We have all sorts of cells that persist for a shorter amount of time than others we might have. For instance, blood cells only last about 100 days and change. Cells of our digestive tract, as you can imagine, that's a very hostile environment. You just imagine the lining of the stomach. We've got all sorts of cells involved in that lining, but they're exposed to these very acidic conditions, and so individual cells there in those tissues don't tend to last a long time, and they are then replaced by division that brings about new daughter cells. So that's something to consider too. So mitosis is really for growth and development, cell replacement, and of course tissue repair, which again is cell replacement. If you damage a tissue, if we lose tissue, that can be replaced as well by cells that will be spurred to divide and produce more daughter cells. So again, if we look at the 2 major stages, we've got interphase and mitosis. Interphase consists of G1, S, and G2, and then we have mitosis consisting of prophase. Again, I've added pro-metaphase, not indicated on the diagram, metaphase, anaphase, telophase. Here, they're saying 1 to 3 hours. Again, highly dependent on the cell you're talking about, but this gives you, an idea of the relative amount of time required for each of these stages. 8 hours or more, 6 to 8 hours, 2 to 4 hours, and so on. So interphase is much longer, as you can tell. There's normal cell function, mostly through G1. G2 might actually be even shorter or almost altogether absent. DNA replication takes some time. That's occurring in S phase. So when you look at G1, S, and G2, for us, this is pretty much what I want you to know, is just what's on this diagram for interphase. We never do discuss that too terribly much, but with interphase, again, most of the cell cycle, it's not actively dividing, consists of G1 DNA synthesis during S phase, and then G2. G1 is characterized by cell growth, might last for 10 hours or so. There's organelle synthesis. You've got lots of protein synthesis going on, and again, you've got the normal functions of the cell occurring. Those functions vary by cell type. So when it comes to the S phase, I definitely want you to realize that DNA is duplicated here. We talked about DNA replication. When does that occur? Right here, during S phase. So when we're talking about topoisomerase and helicase and primase and DNA polymerase, all of that during the copying of DNA, this is where that's happening. It's happening in the nucleus again. That's where your DNA is housed in a eukaryotic cell, and the entire set is copied, and hopefully with great fidelity, meaning that it is an exact copy that we're after here. When we look at, again, the DNA replication forks and look back at that process, that's what we're talking about. G2, additional, quick growth. The cell is going to begin to elongate a little bit because that is going to make for daughter cells that have a size more similar to that of the parent cell. If you think about a parent cell staying the same size and dividing in half, you would expect each daughter cell to be half as big as the original. But what happens is you do see an expansion of the parent cell prior to division, and then of course right after division each daughter cell grows a bit to take on the typical size of that given cell, whatever it might be. But we've got a little bit of protein synthesis. This might be a stage that's rather short or lacking. We'll talk about molecular checkpoints later, but it's fairly important, as you can imagine, that mitosis be regulated. We did mention how that can result in unregulated cell growth results in cancer. Tumor is additional tissue that has developed from cell division that was uncontrolled. New cells that are not supposed to exist, essentially. So that is something of importance to consider. When we look at mitosis, you've got all the phases of cell division that really are focused on what's going on with the nucleus and what's going on with the DNA that's in the nucleus. Why do I say nuclear DNA? Remember that we've got DNA in our mitochondria as animals, and in a plant cell, and plants also undergo, their tissues also undergo, have cells rather, that undergo mitosis. So mitosis is not something that is only occurring in animals. It's occurring across the eukarya, in plants, in fungi, and so on. And when mitosis occurs, we're really focused in again on what's going on with the DNA that's in the nucleus. But plants, I started to say, have DNA in their mitochondria and in their chloroplast. So DNA is found in at least 3 places in a plant cell, 2 places in our cell. And when it comes to prokaryotes, the bacteria, the archaea, really important, really large, diverse groups. Remember that prokaryotes do not have a nucleus, and I'll say right now that they are not performing cell division. They're not dividing by mitosis. There is a separate process called binary fission that we will cover separately for those characters. When it comes to mitosis, we're going to see that it involves again, prophase, prometaphase, metaphase, anaphase, telophase, and that it's followed by cytokinesis, which is the final division of the cell. So I'll stop this video, lead you into a second one that reviews mitosis or covers mitosis and the stages therein.